All right, so today's class, we're going to finish up 6.1. Today we're going to look at proteins and nucleic acids. So proteins, these are complex molecules composed of single units of amino acids. So long chains of amino acids are called peptide chains. And then proteins are made of multiple peptide chains that are folded and bent around each other in a specific shape. So you got the primary structure here, the amino acid sequence. Then we have the secondary structure, tertiary, we see a 3D, we see folding, and then the quaternary structure, which is the complex protein molecule. All right, so a protein, again, it's a chain of amino acids that form the structural parts of cells or act as antibodies or enzymes. And then an amino acid, it's a chemical that contains nitrogen, and it can be linked together to form proteins. All right, so here looking at a structure of a protein, looking at its amino acid structure, it has an amino group right here, the N connected to hydrogens, 2 hydrogens. There's a carboxyl group, COOH. There's the R group, and then there's the hydrogen up here. So the NH2 group right here, that is called the amino group. The COOH, that's the carboxyl group. The R group can be represented in a number of different structures, and it differentiates one amino acid from another. So proteins are composed of 20 different amino acids. So here's just 6 different amino acids: serine, threonine, cysteine, here's glutamine, asparagine, and tyrosine. All right, and you can see that they all have the amino group, the carbon, the hydrogen group, and then the carboxyl group. And then it's this R group that changes and differentiates the 20 different amino acids. Okay, so proteins have peptide bonds, and this is the bond shown here that will bond and join the amino acids together. So once you have a chain of 3 or more amino acids, it's called a polypeptide. So again, it is happening during this dehydration synthesis where there is a removal of water. All right, and an essential amino acid is an amino acid that must be obtained from the diet. So we get this from the foods we eat. All right, so just summarizing the essential nutrients here, carbohydrates are molecules that contain hydrogen, carbon, and oxygen. They are the preferred source of energy for cells. So here sources are, for example, plants. In humans, it is the primary energy source. Lipids are formed from glycerol and fatty acids. They are energy storage compounds. Recall that plant oils are the unsaturated fats, easier to break down. Animal fats are saturated fats and are much harder to break down. Functions in humans included energy storage, insulation of skin, cushioning the organs, as well as synthesis of hormones and steroids. And then proteins, what we just looked at, they're molecules, they're made up of amino acids, and they're the structural components of cells. So sources are from plants and animals. They are the structural components of the cell, make up enzymes as well as antibodies. All right, just to review the anabolic and catabolic reactions. So organic molecules, they contain carbon and form the basis of most living things. And it's been estimated that in nature there are millions more organic compounds than there are inorganic compounds. And all 4 of the macromolecules, the carbohydrates, lipids, proteins, we haven't looked at nucleic acids yet, they are organic compounds. And each of these molecules is formed from simpler subunits in an anabolic reaction. So the dehydration synthesis is a type of anabolic reaction where we saw the removal of water. Each macromolecule may be broken down again into its original repeating simpler subunits through catabolic reactions. And then in catabolic reactions, molecules are broken down into smaller components and energy gets released. So when we break down food during digestion, that's a catabolic reaction. And that happens through a hydrolysis reaction. All right, it's a type of catabolic reaction. And do note that both of these reactions require enzymes as well to take place. All right, this using indicators is a method common in experiments. A chemical indicator, it's a compound that gives a visible sign indicating a particular type of chemical is present. So for example, like when we did acid bases, you would have used like litmus paper or pH paper. And in Bio 20, you would use acid base indicators as well as some of these new indicators to indicate the presence of a specific macromolecule. All right, so remember that acid base indicators, they changed color depending on the pH range. Here we're looking at some common indicators used to test for macromolecules. So if you're testing for fats or lipids, there's an indicator called Sudan 4. Its positive result will show a red-orange color, and if it's a negative result, it will be a dark red color. To test for proteins, there's an indicator called the Buret reagent, and a positive result will be violet or black, a negative result will show blue. To test for simple sugars, there is an indicator called the Benedict solution, and there's a range of colors here. If it's green or yellow, it shows a low concentration of simple sugars. Yellow or orange is a high concentration, and if there's an orange to red color, it shows a very high concentration of simple sugars. The negative result will stay as blue. To test for starch, the indicator used is iodine. The positive result will be blue-black. The negative result will be a brown color. All right, so here are just some diagrams and pictures showing, for example, here iodine, the positive result on the left here, and then we see the negative result on the right. Here's the test using the Buret reagent to test for proteins, and it's a positive result. Again, positive for Buret was a violet color or black. So that's a positive result. And then this was the results for Benedict's test for sugars. So again, simple sugars, there was this range of colors. All right, so green or yellow, it's a low concentration. And as we get to the orange and reds, it becomes a higher concentration. And if it's blue, it's a negative result. So here, a blue would show there's a negative result for those sugars. All right, there is a video we'll watch here on biomolecules. We'll just watch it at the end here. Enzymes, recall that enzymes are made up of proteins. So there's specific types of proteins that help regulate the many different chemical reactions that occur in our bodies. So enzymes, they lower the activation energy required to catalyze reactions. Otherwise, the energy required would be too large, and these reactions could cause damage to the body cells. So enzyme function can be simply described as speeding up of a chemical reaction due to the interaction of an enzyme's active site with a specific substrate molecule or molecules. So for example, if we're looking at the enzyme lactase, it's only going to bind to the substrate molecule of lactose. Hydrolysis Just to review that it's the decomposition of a compound by a reaction with water. So the hydrolysis of sucrose into its components of glucose and fructose is controlled by the enzyme sucrase. It's found in the human digestive tract. So there is a video here on enzymes. It's another Amoeba Sisters video on enzymes. All right, and then in the video we'll see that the enzyme never actually becomes part of the reactants or products. It can be used over and over. One factor that affects enzyme action is inhibition, and I'll show you how the end products of a series of enzymatic reactions will inhibit enzyme activity in a negative feedback loop. So feedback inhibition ensures that the body doesn't produce more products more than is needed. It ensures that the body is only producing as much as required. All right, so here looking at enzymes, here we have an enzyme and its substrate, this active site right here are available. Here is the substrate, which is the maltose, and it's going to bind together. So the substrate binds to the enzyme to create this enzyme substrate complex. All right, so the active site here, right here, it's the area of an enzyme that combines with the substrate. The catalyst, it's a chemical that increases the rate of chemical reactions without altering the product or being altered itself. The enzyme is the protein catalyst. So this blue structure here in this example, it permits chemical reactions to proceed at low temperatures. And then the substrate here, the molecule on which an enzyme works. All right, so this is just another diagram showing that we have a substrate, sucrose, which is made up of glucose and fructose bonded together. Here's the enzyme. Here is its active site. They will bind together. The substrate binds to the active site of the enzyme, sucrase. Water is added. It's a hydrolysis reaction. The bond between glucose and fructose gets broken, and we have our products released. Glucose and fructose get released, and the enzyme is then free to go bind to another sucrose substrate. All right, so factors that affect enzyme action. Number one is pH, acidity. Number two is the substrate molecule concentration. So the greater the number of substrate molecules, the greater the number of collisions, and the greater the rate of the reaction. So here in figure 3 and 4, figure 3, different enzymes function within different pH ranges. So for example, the stomach is very acidic, so it's going to function at about pH of 2. And then in the intestine is not acidic, it becomes more basic. And the trypsin works at that pH of about 8. And then figure 4 is showing the reaction rate begins to level off at point X because there is a limit to the amount of enzyme that's available. Lastly thirdly, the third factor that affects enzyme action is temperature. So here you can see temperature on the x-axis, the reaction rate on the y-axis. Typical human enzyme at about body temperature, which is about 37 degrees Celsius. And then this is a bacteria. It's a thermophilic or heat-tolerant bacteria, and we can see that its optimum is close to 80 degrees Celsius. A fourth factor that affects enzyme action is competitive inhibition. So competitive inhibitors have shapes that are very similar to that of the substrate, and they will compete, the inhibitors compete with the substrate molecules for the active site of the enzymes. All right, if the inhibitor remains joined to the substrate or joined to the enzyme, the substrate cannot bind. And it's a little bit blurry here, but remember it will be clear when you open up the PowerPoints under modules. All right, so figure A here showing that the substrate normally binds to this active site, but a competitive inhibitor, this reddish one here, competes with the substrate for the active site. All right, and as the product from a series of chemical reactions begin to accumulate within a cell, the product itself interferes with one of the enzymes in a process known as feedback inhibition. And the interference will slow down the reaction rate, preventing the accumulation of final products. The final product of the metabolic pathway interferes with the enzyme by combining with its regulatory site. This alters the active site and prevents the union of the enzyme and the substrate. All right, so here in this diagram, the substrate binds to the active site of the enzyme. There's this area here called the regulatory site. So the final product from this reaction will attach to this regulatory site here, and it'll change the shape of the enzyme. So notice how this structure has changed so that the substrate can no longer bind. All right, and that will prevent, or it'll slow down the production. All right, so carbohydrates, lipids, proteins, and nucleic acids are macromolecules. They all have distinct characteristics. Carbohydrates can be simple sugars or polysaccharides. Their purpose is to store energy. Lipids are insoluble in water. They store energy. They form hormones like estrogen and testosterone. They also comprise membranes because of their insoluble nature. The bond structure of lipids causes them to be saturated or unsaturated. Proteins are the most functionally diverse macromolecule. Hair, bones, muscles, nuclei, nails, they all contain different proteins, and proteins are made up of chains of amino acids. An amino group is connected to a carbon molecule, which is bonded to an acid group. There's 20 different kinds of amino acids that can bond together in different combinations to give different proteins. The physical nature of proteins determines whether they're going to be water soluble, like enzymes, or insoluble, like fingernails. Then enzymes are designed to bind to substrate molecules, which are any macromolecules. Each enzyme has a specifically shaped binding or that activation site, and enzymes are there to speed up reactions in building or breaking down molecules. If we didn't have enzymes, we wouldn't be able to quickly access the stored energy of glucose from a polysaccharide chain, and as a result, the cells would die. Enzymes require optimal conditions to work. So, for example, pH, temperature, were a couple of factors for enzymes to work optimally.